biotechnology deals with techniques of using live organisms or enzymes from organisms to produce products and processes useful to humans. In this scene, in this sense, okay, biotechnology deals with techniques of using live organism or enzyme from organism to produce products and processes useful to humans. In this sense, making curd, bread, or wine, which all which are all microbe-mediated process, could also be thought as used in a restricted sense to those processes which use organism to achieve the same on the large scale. Further, many other processes techniques are also included under biotechnology. For example, in vitro fertilization leading to a test tube baby, synthesizing a gene and using it, developing a DNA vaccine, or correcting a defective gene are all parts of biotechnology. The European Federation of Biotechnology EFB, European Federation of Biotechnology, has given a definition of biotechnology that encompasses both traditional view and modern molecular biotechnology. The definition given by EFB is as follows: the integration of natural science and organism, cell parts thereof, and molecular analogues for products and services. Principles of biotechnology. Among many, the two core techniques that enable birth of modern biotechnology are genetic engineering, techniques to alter the chemistry of genetic material, DNA and RNA, to introduce these into host organism and thus change the phenotype of the host organism. Maintenance of a sterile, microbial, contamination-free ambience in chemical engineering processes to enable growth of only the desired microbe eukaryotic cell in large quantities for the manufacture of biotechnological products like antibiotics, vaccine, enzymes, etc. Let us now understand the conceptual development of the principle of genetic engineering. You probably appreciate the advantages of sexual reproduction over the asexual reproduction. The former provides opportunities for variation and formation of unique combination of genetic setup, some of which may be beneficial to the organism as well as population. Asexual reproduction preserves the genetic information while sexual reproduction permits variation. Traditional hybridization procedures used in plants and animal breeding very often leads to inclusion and multiplication of desirable gene along with the desired gene. The technique of genetic engineering, which includes creation of recombinant DNA, use of gene cloning, gene transfer, recombinant DNA, use of gene cloning and gene transfer, overcome this limit and allow us to isolate and introduce only one of the set of desirable gene without introducing undesirable gene into the target organism. Do you know that do you know the likely fate of piece of DNA which is somehow transferred into an alien organism? Most likely, this piece of DNA would not be able to multiply itself in the progeny cell of the organism, but when it gets integrated with the genome of recipient, it may multiply and be inherited along with host DNA. This is because of the alien piece of DNA has become part of chromosome, which has the ability to replicate. In chromosome, there is a specific DNA sequence called the origin of replication, which is responsible for initiating replication. Therefore, for multiplication of any alien piece of DNA in an organism, it needs to be part of chromosomes, which has a specific sequence known as origin of replication. Origin of replication. Origin of replication. DNA, recombinant DNA, gene cloning, gene transfer. Recombinant DNA, use of gene cloning, gene transfer. Origin of replication. Okay. origin of replication. Specific DNA sequence. In chromosome there is a specific DNA sequence called the origin of replication, which is responsible for initiating replication. Therefore, for the multiplication of an alien piece of DNA in an organism, it needs to be a part of chromosome, which has a specific sequence known as origin of replication. Thus, an alien DNA is linked with the origin of replication so that this alien piece of DNA can replicate so that this alien piece of DNA can replicate and multiply itself in the host organism. This can also be called as cloning or making multiple identical copies of any template DNA. This can be called as cloning or making multiple identical copies of template DNA. Origin of replication, cloning or making multiple identical copies of DNA. Let us focus on the first instance of construction of an artificial recombinant DNA molecule. The construction of the first recombinant DNA emerged from the possibility of linking a gene encoding antibiotic resistance with a native plasmid, autonomously replicating circular extrachromosomal DNA of Salmonella typhimurium. Stanley Cohen and Herbert Boyer accomplished this in 1972 by isolating the antibiotic resistance gene by cutting out a piece of DNA from plasmid, which was responsible for conferring antibiotic resistance. The cutting of DNA at a specific location became possible with the discovery of the so-called molecular scissor restriction enzymes. The cut piece of DNA was then linked with the plasmid DNA. The plasmid DNA is a vector to transfer the piece of DNA attached to it. You probably know that the mosquito act as insect vector to transfer the malaria parasite into human body. In the same way, a plasmid can be used as a vector. Used as a vector to deliver alien piece of DNA into the host organism. Linking of antibiotic resistance gene with plasmid vector become possible with the enzyme DNA ligase, which acts on cut DNA molecules and joins the end. This make a new combination of circular autonomously replicating DNA created in vitro and is known as recombinant DNA. When this DNA transfers into Escherichia coli, a bacterium closely related to Salmonella, it could replicate using the new host DNA polymerase enzyme and make multiple copies. The ability of multiple copies of antibiotic resistance gene in E. coli was called cloning of antibiotic resistance gene in E. coli. Three basic steps in genetically modifying of an organism. Modifying an organism. You can hence interfere. There are three basic steps in genetically modifying an organism: identification of DNA with the desirable gene, introduction of the identified DNA into the host, maintenance of the introduced DNA in the host, and transfer of the DNA to its progeny. Identification, introduction, and maintenance. Identification, introduction, and maintenance. Identification, introduction, and maintenance. Okay. Alter the chemistry of genetic material. Change the phenotype of host organism. Maintenance of sterile, microbial contamination-free ambience in chemical engineering. I don't like it. Let's end this one.